Welcome to the first video on photosynthesis that will cover a little bit of background information on the process as well as the light reactions. We will save the Calvin cycle and all of its detail for a second video. At this point, you should be intimately familiar with the reactions of both cellular respiration and photosynthesis. But at this point, I want to remind you that in cell respiration, we were dealing with the oxidation of glucose, so as to extract energy in quantized amounts and use that energy to synthesize ATP. In photosynthesis, we're not performing oxidation, and in fact, it probably won't surprise you that instead we're going to be producing carbon dioxide, adding electrons to carbon intermediates with the ultimate goal of synthesizing carbohydrates like glucose. So you've seen this slide before, and it should be familiar to you that there's an interplay between photosynthesis and cell respiration. Of course, if you recall, the reactants of cell respiration are also the products of photosynthesis, and similarly, the products of cell respiration are the reactants for photosynthesis. In general, what I do want to remind you of is that plant cells have both of these organelles. It's a very common misconception to forget about the fact that plant cells have both chloroplasts and mitochondria. Of course, animal cells, fungal cells, only have mitochondria. Some quick review as to some of the terminology that we may be using over the course of this unit and over the course of the rest of the year. Remember that autotrophs are your producers, your lowest trophic level organisms, while heterotrophs are your consumers, and typically we organize consumers by the level at which they feed. So if you remember from 10th grade, there are first level consumers, second level consumers, and so on. Producers are going to have the means to acquire energy from inorganic materials. So when I write here that the cell feeders are acquiring energy from sunlight, of course that's a transformation of light energy to chemical potential energy. And heterotrophs, of course, are going to have to acquire the energy by consuming or ingesting some sort of product, and then digesting that product that we've seen earlier in the year, absorbing those molecules across membranes, and then using those biological molecules to build other more complex biological molecules in anabolic processes. So at this moment, what I'd like you to do is to consider the reaction for photosynthesis. You'll see that in this reaction, atoms of oxygen appear on both sides of the equation, and that they appear in both the reactants and the products. So what I'd like you to do is consider an experiment that you could design that would determine the source of oxygen in a molecule of glucose. As you do this, think about some positive and negative controls that you might include. Any good experiment includes these two controls. And then also consider which molecules that are serving as the reactants in this process are ultimately going to be oxidized, and which ones are going to be reduced to generate the products glucose and oxygen. So take a moment, grab a piece of paper or your notebook, and see if you can highlight or at least sketch out an experiment that you might perform. Pause the video now until you complete that task. So what did you come up with? Did you consider using radioactive isotopes of any of the elements in the reactants? Hopefully you did. If you consider labeling the carbon dioxide with heavy oxygen, that might be O18, versus labeling the oxygen in water with heavy oxygen, again, O18, you can actually trace the path of those oxygen atoms through photosynthesis. And what we find is that the oxygen atoms in water are ultimately going to serve as the oxygen atoms that give rise to molecules of oxygen produced at the end of photosynthesis, where the oxygen in carbon dioxide will ultimately give rise to the oxygen in molecules of glucose. So hopefully you were able to come up with an experiment that could test that hypothesis. Well, if we begin to look at photosynthesis in more detail, we can divide the process into two general sets of reactions: the light reactions and the Calvin cycle. Again, we're going to focus on the Calvin cycle in another video. But in general, just to highlight the key differences, in the light reactions, molecules of water are going to be split. Now when I say split, ultimately what that means is that electrons will be removed from molecules of water, and this will be the step where oxygen is produced. This, of course, is going to be a oxidation step in the case of water. Remember, our ultimate goal is to reduce molecules of carbon dioxide through photosynthesis. The light reactions are going to produce a molecule of NADPH. This is actually really similar to NADH. The only difference between the two molecules is the presence of a phosphate group in NADPH. Okay. We're going to generate some ATP via a process known as photophosphorylation, which really is not that drastically different from what we saw in cell respiration and oxidative phosphorylation. I do want to remind you that the light reactions do not produce any sugar or sugar intermediates. Really, the only products of the light reaction are NADPH and ATP. And the light reactions are going to occur on the membrane of the thylakoids, those membranous stacks on the inside of the chloroplast. In contrast, the Calvin cycle, this will be the carbon fixation step. This is where carbon dioxide, molecules of CO2, will be reduced into simpler carbon molecules, I should say more complex carbon molecules, like G3P. I say simple because many people consider G3P to be an intermediate of glycolysis. That's something that you should consider. But G3P certainly isn't glucose. So when I say simpler, I'm really referring to G3P as a simpler intermediate on its way to glucose. The molecules of NADPH and ATP produced during the light reactions are going to be used in the Calvin cycle to, again, not surprisingly, reduce molecules of carbon dioxide. Sometimes you'll see the Calvin cycle referred to as the dark reactions or the light-independent reactions. I don't want you to think that the Calvin cycle only occurs at night and the light reactions only occur during the day. In fact, both of these processes can occur during the day. Of course, the Calvin cycle could only take place at night using stored NADPH or ATPH generated during the day by the light reactions. The Calvin cycle, in contrast to the light reaction, will take place in the stroma of the chloroplast, and again, we're not going to produce glucose directly through the Calvin cycle. This can be very misleading when you look at textbook diagrams. Instead, we're going to produce molecules of G3P that later on in a biochemical pathway will be linked together to form a molecule of glucose. In a way, you can think about this process, the conversion of G3P to glucose, as the reverse of glycolysis. So this figure summarizes both the light reactions and the Calvin cycle. Starting with the light reactions, we can see here is the chloroplast. Here are the grana, the stacks of membranous thylakoids. Each individual sac is a thylakoid. So photosynthesis will take place on the membrane of the thylakoid. And the light reactions are going to generate ATP and NADPH, as we saw earlier, as well as molecules of oxygen. The Calvin cycle will reduce carbon dioxide, but will use the products of the light reactions to reduce CO2 to molecules of G3P and ultimately to glucose. And of course, these two processes are interconnected, so the products of the Calvin cycle, i.e. The oxidized form of NADP and the dephosphorylated form of ATP, will be recycled back to the light reactions, and as we've seen now numerous times, molecules of ATP and NADPH are going to be produced and then used by the Calvin cycle. So again, we're going to be focusing on the light reaction in this video. At this point, you can really think of oxygen as a waste product of photosynthesis, as well as the simple carbohydrate intermediates. But remember, plant cells have mitochondria too, so the oxygen and the carbohydrate that's produced through these processes will be used by plant cells themselves. Of course, they can't use it all. If they did, there would be no net gain of carbohydrate or oxygen within an ecosystem, and organisms at a higher trophic level would cease to exist. So it's important that these plant cells don't use everything that they made. If they did, well, we as higher level consumers would be in deep, deep trouble. Alright, so before we go into the finer details of the light reactions, it's important to go back and review briefly the structure of a leaf, because of course if we're dealing with photosynthetic organisms, we're dealing with the site of photosynthesis, and that often is going to be the green leaf. If we look at a cross-section of a leaf, you're probably familiar with the structure of stomata. These are the pores along the leaf in which gases will freely be able to move into or out of the plant. So typically if we're dealing with photosynthesis, we're dealing with CO2 entering and O2 leaving. If we were to zoom in on a particular location of the leaf, that being the mesophyll, we'd find individual mesophyll cells that are jam-packed full of chloroplasts. Within the chloroplasts that we've seen on the previous slide, we're going to have our stacks of membranous thylakoids. Those are the grana, and the site of photosynthesis will be, of course, a thylakoid membrane. And the fluid in which all these structures are suspended is the stroma. You can think of the stroma as the cytosol of the chloroplast. Again, another look at a cross-section of a leaf. We're focusing primarily on the mesophyll cells. These are the chloroplast dense cells of the leaf in which photosynthesis will take place. Yet another look at the microscopic architecture of the inside of a chloroplast. Again, here we have our granum, plural, multiple granum are your grana, and we're focusing again on the fact that the light-dependent reactions are occurring on the membrane of the thylakoid. If we're talking about photosynthesis, it's important to review briefly the electromagnetic spectrum. We're going to focus primarily on the small subset of the electromagnetic spectrum that makes up visible light. This ranges from 400 to approximately 700 nanometers. Of course, there are other wavelengths of light that are important to chemists and physicists, but to biologists, really, it's only the visible light spectrum that is crucially important to us. Quick reminder that if you're dealing with a wavelength of light that is small, that means its frequency is high, and of course that means it has a great deal of energy. If you're dealing with a wavelength of light that is long, its frequency, of course, is going to be smaller and will carry less energy per photon. So you hopefully at one point have asked yourself why certain objects have color in the first place. This is going to be important to us because when we say things like leaves are green, do we actually know why leaves are green? Well, the reason why objects have any color whatsoever is because wavelengths of light within that visible light spectrum are either reflected or transmitted by objects. So it's only light that's absorbed that has the capacity to be used in photosynthesis. And in fact, it's absorbed light that's going to be invisible to the human eye. We really don't have a means to detect that light if it's being absorbed by an object. We're really focusing on whether or not light has been transmitted through the object or if light has been reflected off of the object. So leaves are green because green light ultimately is going to be either transmitted or reflected by the molecules in the leaf, while other wavelengths of light are going to be absorbed. And it's important to begin to appreciate which wavelengths of light are going to be absorbed in photosynthesis and which wavelengths of light are going to be reflected. So what you see here is a classic electromagnetic spectrum of a leaf. In fact, we're going to be doing this in lab. We'll take some spinach leaves or other leaves that you may have lying around your house. We'll extract the different photopigments from those leaves, place that extract into a spectrophotometer, and run a full absorbance spectrum analysis on that sample. And what we'll probably see is something very similar to what you see here, where the photopigments that are capable of absorbing light in photosynthesis actually appear in a mixture. They appear as a mixture of chlorophyll A and chlorophyll B, along with other accessory pigments, which are often referred to as the carotenoids, which have the capacity to absorb light at different wavelengths. So you'll see here that chlorophyll A, for example, has a maximum absorption wavelength at around 450 nanometers, while it has a secondary absorption peak at around 680 nanometers. Chlorophyll B, again, two peaks, one around 470 and a second right around 650, and then you have these carotenoids and other molecules that are known as the xanthophylls, which also have the capacity to absorb light, but they absorb light, as you can see, at slightly different maxima compared to chlorophyll A and chlorophyll B. Notice down here where the green wavelengths would appear that there is very little absorption either by chlorophyll A, chlorophyll B, or any of the carotenoids, because again, those light wavelengths are going to be reflected or transmitted and not absorbed for the purposes of photosynthesis. Okay, so how does a thylakoid, how does a chloroplast, how does a plant cell actually perform photosynthesis? Well, in order to understand this process, we have to really get into what a photosystem is. And a photosystem is going to be the unit in which a photosynthetic process can take place. So if you go back to that picture of the thylakoid and begin to picture that a thylakoid membrane may be covered with thousands of photosystems, we're going to be able to divide those photosystems into two parts, photosystem 1 and photosystem 2. And these photosystems are networks of chlorophyll molecules, accessory pigments, again those are your carotenoids and xanthophylls, and other proteins that are going to be at the surface of the thylakoid. And what we'll see is that these photosystems ultimately are going to collect light through antenna complexes. So that's going to be a collection of molecules that will be capable of absorbing light from the surroundings and then transmitting that light to what's known as a reaction center. And the reaction center will contain one or more chlorophyll molecules. So this in general is what the photosystem would look like, where a photon of light would be absorbed by an accessory molecule. This could be a carotenoid, it could be a xanthophyll, it could even be a chlorophyll B molecule. And the energy is going to be transferred within these molecules of the photosystem all the way to the reaction center. If we look at the molecules that make up that thylakoid, here's a typical molecule of chlorophyll, and if you'll just briefly notice its structure, it has a long hydrocarbon chain that's going to allow it to embed in the membrane, and then this porphyrin group, which ultimately is responsible for absorbing light at the surface of the molecule, or at the surface of the membrane. All right, so back to the structure of the photosystem and how it works. Again, we have our antenna complexes, which ultimately will absorb light and then transfer energy from chlorophyll molecule to chlorophyll molecule all the way to this reaction center. And what we're going to want to do is transfer that energy in a way so that ultimately electrons at the reaction center can be elevated to a higher energy state and then picked up by an electron acceptor, so that ultimately the energy stored in that electron can be used to do work within the cell. That work that, as we'll see, is going to be very familiar. It's going to be the generation of a proton gradient, which is going to be utilized in ATP synthesis. Here's a diagram of the photosystem again. Here's your photon of light. Here are your chlorophyll complexes, which ultimately are going to transfer their energy all the way to a special group of chlorophyll molecules. Electrons within that chlorophyll molecule are going to be elevated to a higher energy state, and then there'll be some molecule that will pick up that electron and transfer it down to a lower energy state, allowing the cell to do work in the process. Well, at this point you may be asking yourself, how is the energy transferred from chlorophyll complex to chlorophyll complex within this photosystem? And actually, you've seen a mechanism in the past that can be used to explain how the energy is being transferred from molecule to molecule. A lot of textbooks don't really delve into this topic, and I think it's important that we actually spend a minute or two on it so that you can appreciate just how elegantly simple the mechanism is. If you're not careful, you're going to begin to think that light energy will be absorbed by an antenna complex within that photosystem, and that somehow light energy jumps from chlorophyll complex to chlorophyll complex. And that's not at all what happens. Some people would also argue that when those antenna complexes absorb light, that heat will be generated, and that heat will be transferred from molecule to molecule on its way to the reaction center. That's not what happens either. Instead, what happens is resonance energy transfer. And we've seen this when we talked about fluorescent resonance and energy transfer. So, if you think about the molecules that are absorbing light, when they do so, they'll elevate electrons to a higher energy state, and that's going to cause molecules to vibrate. That vibrational energy, assuming that the neighboring molecule is in close proximity to the excited molecule, will be transferred to the molecule with less potential energy, and that energy transfer will cause one electron or one molecule to fall back down to ground state, while another molecule becomes excited. So we have resonance energy transfer because the molecules are wiggling, and because they wiggle, the energy that is present in that molecule will cause another molecule to wiggle, and then when that molecule wiggles, it will cause electrons to become elevated to a higher energy state and thus transferred from one molecule to the other. Okay, so I want to show you this process using one of our creepy virtual cell videos, but I'm not going to show you the entire video. I can put a link up to the website if you want to watch the entire video in all of its glory. So I'm going to go ahead and get the video started, and then pause it a little bit along the way. So just to show you the energy transfer, I'm going to start the video right here.
These proteins provide a way to harness the unique energy contained in light. When one of the many photons of light flooding a leaf hits a chlorophyll molecule surrounding the reaction center, it creates resonance energy. You can think of this as vibrational energy. Here you can see the chlorophyll vibrating. That resonance or vibrational energy is then passed to a neighboring chlorophyll molecule. It is then passed through several chlorophyll molecules until it reaches the P680 reaction center. It is that energy that results in the loss of an electron from the P680 molecule.
Okay, so now that you've seen the energy transfer in action via that animation, we can again look at the photosystem in a little bit more detail, and we can begin to add some levels to our understanding. So again, here's the light striking that antenna complex. See that pretty clearly here. The energy will be transferred from chlorophyll to chlorophyll molecule by resonance energy transfer until we get to that reaction center, which contains a special class of chlorophyll molecules. These are classified as P680. That's just because that is their maximum absorption wavelength. And as the energy is transferred, it's going to cause an electron to become elevated to a higher energy state and then be accepted by another molecule in the path. Okay. In that process, of course, as electrons become elevated to a higher energy state, they have to be replaced by electrons at that reaction center. Otherwise, as additional photons of light come through, there won't be an electron to be transferred to that higher energy state. Well, at this point, we can go back to the idea that water is being oxidized, and as water is oxidized, oxygen is produced, and it's that oxidation of water that ultimately will produce the electrons, which provide the reaction center of this photosystem with the electrons that are necessary to be elevated to the next higher energy level. Okay, so at this point, things should start to look kind of sort of familiar. After the electrons have been elevated to a higher energy state, they're passed down our good friend, the electron transport chain. Now this chain is different than the one found in the mitochondrion, but in the grand scheme of things, it's not that drastically different. PQ stands for plastoquinone. You can think of that as an analog to ubiquinone. And PC is plastocyanin. You can think of that as an analog to cytochrome C. And then you have an additional cytochrome complex, which is going to move protons across a membrane, and that proton gradient will ultimately be used in ATP synthesis. As those electrons fall down the electron transport chain, they will make their way to a second photosystem. This is photosystem 1. And notice that we have photosystem 2 followed by photosystem 1. The numbering that we have here has nothing to do with the order in which the electrons are moving. In fact, photosystem 2 is named 2 because it was discovered second. As we see here, the electrons will fall back down the electron transport chain, the region of the reaction center. That's okay, because light will be transferred through resonance energy transfer to that reaction center. That will elevate electrons to a higher energy state. So we can think of the electrons that are moving down the chain as replacing the electrons that are being elevated via the energy transfer, again, to another accepting molecule. And in this case, the final electron acceptor of the photosystem 1 will be a molecule called ferredoxin. And ferredoxin will ultimately deliver its electrons to another enzyme complex, NADP plus reductase, because again, we're going to be reducing NADP plus to NADPH. Remember, the P stands for a phosphate group, but you can think of NADPH as being virtually identical to NADH, as that we saw in cell respiration. And this will be the source of the NADPH in the light reactions, and of course the ATP has been generated via the creation of the proton gradient across the thylakoid membrane, and the gradient will be used in ATP synthesis via an enzyme, not surprisingly, known as ATP synthase. So here we can see the process all together. We can see photosystem 1, electrons being elevated to a higher energy state, those electrons being replaced via the oxidation of water and the production of oxygen. The electrons fall down the electron transport chain in the process generating a proton gradient that will be used in ATP synthesis. The electrons that fall down will eventually reach photosystem 1. Those electrons that are being elevated via additional absorption of photons will be replaced by the electrons coming down the electron transport chain. Those elevated electrons will be passed to ferredoxin, and then ferredoxin will ultimately transfer the electrons to NADP+. Again, another look at the combinations of photosystem 1 and photosystem 2. I encourage you to focus on where NADPH and where ATP are going to be generated. I don't believe that the AP examiners are going to ask you this anymore, but in the past, they used to have students focus on the maximum absorption spectra of photosystem 2 and photosystem 1. Of course, as you can see here, photosystem 1 has a maximum absorption spectrum of 700 nanometers, while photosystem 2 has an absorption spectrum of 680 nanometers. I don't think that's going to be as critically important as it has been in years past. Alright, so we're almost done with the light reactions, and in order to further understand the generation of that proton gradient across the thylakoid membrane, I thought it would be useful to briefly go back to cell respiration and the generation of the proton gradient across the inner mitochondrial membrane. You know that protons ultimately are being pumped from the matrix across the membrane into the intermembrane space within the mitochondrion. In the case of the chloroplast, you can think of the protons instead being pumped from the stroma into the thylakoid space. So think of the thylakoid membrane as the inner mitochondrial membrane, okay? And then as those protons move up into the thylakoid space, they will eventually move back down through that thylakoid membrane, through an ATP synthase, and that proton gradient's energy harnessed to synthesize ATP. So this picture shows that process in a little bit more detail. We can see our photosystems. Here's photosystem 2, delivering electrons to the first electron acceptor in the electron transport chain, that's plastoquinone. In plastoquinone, as it delivers electrons to subsequently lower and lower energy states, the energy that's being released will be used to move protons across the membrane. Again, this is the thylakoid membrane, so protons are moving from the stroma into the thylakoid space. That will generate a proton gradient within the thylakoid that ultimately will be used via an ATP synthase to produce ATP. Again, this picture probably shows this process in the most detail. So just one more time, here we can see our photosystem 2. Light strikes the photosystem, energy transfer, electrons are elevated, replaced by water, fall down the chain, protons are pumped across the thylakoid membrane, proton gradient is generated, electrons continue to move down the chain until they reach photosystem 1, photon strikes, elevates electrons, electrons replace those elevated electrons, electrons are passed from ferredoxin to NADP plus reductase so as to produce NADPH. The proton gradient generated through that electron transport chain will be used to make ATP. Both NADPH and ATP are going to be generated within the stroma of the chloroplast, and as we said at the beginning of the video, those molecules will be important precursors, important substrates in the reduction of carbon dioxide during the Calvin cycle. There are going to be times, though, where the non-cyclic electron flow that we have seen now for the past few slides is not going to be utilized by the chloroplast. And under those circumstances, the chloroplast will use photosystem 1 exclusively, but not photosystem 2. And this is going to happen when ATP is being used faster than it's being produced. So, in essence, during the cyclic electron flow, where photosystem 2 is bypassed, so you can see that it's blocked out over here, we're going to exclusively use photosystem 1. So again, light will be absorbed. Okay, the light will be used to elevate electrons to a higher energy state, and those electrons will be passed on to ferredoxin. But rather than those electrons being passed from ferredoxin to NADP plus reductase, thus yielding NADPH, instead it's possible for the chloroplast to divert those electrons from ferredoxin back to the electron transport chain, so as to further enhance that proton gradient and further increase the production of ATP. This is only going to happen when ATP is needed. For some reason the cell is in desperate need of it, but it is not in desperate need of the reduced form of NADPH. Just one more look at the exact same process, perhaps in a little bit more detail. Cyclic electron flow and how it differs from non-cyclic electron flow. Alright, so that's it. I know this has been a long video. I thank you for sticking with it, and hopefully you found it helpful. If you have additional questions, please don't hesitate to contact me by email or come see me before or after class.